Hello student, good morning. Today we will start the new topic, that is linkage. What is meant by linkage? An organism often has many phenotypic traits in its body because each phenotypic trait of an organism is determined by at least a pair of alleles, genes, which reside at specific gene loci of homologous chromosomes, autosomes or sex chromosomes. Therefore, it can be expected that an organism may have numerous genes for its various phenotypic traits. The number of genes of an organism thus may exceed the number of its chromosomes because commonly most organisms contain limited number of chromosomes. For instance, man has thousands of pairs of genes but only 23 pairs of chromosomes. Hence, each chromosome must contain a large number of genes. Further, because the chromosomes are inherited as units, that is, they pair and segregate in meiosis as units to gametes, so all of the genes which may specify various different phenotypic traits but are located in any given chromosomes tend to be inherited together. All the genes on the chromosomes are said to be linked to one another and belong to the same linkage groups. The phenomenon of inheritance of linked genes in same linkage group is called as linkage. Difference in linkage and independent assortment. Mendel's law of independent assortment is applied only to those genes which are located on separate chromosomes, because the linked genes of a linkage group chromosome inherit together. A dihybrid contains either linked genes or independently assorted genes can be determined by test crossing it with a double recessive parent. The independently assorted give the test cross ratio of 1:1:1 and linked genes give the test cross ratio 1:1, as have been illustrated by the following examples. If genes occurs on different chromosomes, they assort independently and give a test cross ratio of 1:1:1 as follows. Parents capital AA, capital BB cross with small a, small bb. Gametes capital A and capital B, small a and small b. This is the gametes. Test cross. Capital A, capital B, small b. Cross with small a, small b, small b. Gametes capital AB, capital A, small b, small a, capital B, small a, small b. and small a small b. One fourth capital A capital B small b. One fourth capital A small b small b. One fourth small a capital B small a b. One fourth small a small b b, or 1:1:1:1 test cross ratio. Example second, the linked genes do not assort independently but tends to stay together in the same combinations as they were in the parents. In the following figure, the genes on the left of the slash line are on the chromosomes and those on the right are on the homologous chromosomes. The linked genes give the test cross ratio of 1:1 as follows. Parents capital A capital B small a small b cross with small a small b oblique small a small b. Capital A capital B oblique small a small b cross with small a small b oblique small a small b. Gametes capital AB small ab small ab small ab. Half of the small ab oblique small capital AB oblique small ab is to half small a small b oblique small a small b, or 1:1 test cross ratio. Views of the classical geneticist on linkage. Mendel could not notice the phenomenon of linkage because fortunately the 7 pairs of factors or alleles studied by him in P were located on 7 different pairs of chromosomes. It was noticed and discovered by some other post Mendelian genetics who during their genetic investigations came across to linked gene. The evolution of linkage concept look placed by the view of following classical genetics. Sutton's views on linkage. Sutton's in 1903 was the first classical geneticist who made certain predictions about the linkage merely by performing certain cytological investigations. He suggested that each chromosome must bear more than a single gene and that the genes represented by the one chromosomes but inherited together. However, he could not prove his prediction by genetic experiments. Second number, coupling and repulsion hypothesis of the Batson and Punnett. Batson and Punnett, that is the scientist. He discovered in 1905 to 1908 formulated the hypothesis of coupling and repulsion to explain the unexpected F2 results of a dihybrid cross between a homozygous sweet pea Lathyrus odoratus having a dominant alleles for blue or purple flowers, capital RR, and long pollen grains, capital R, zero and capital R 0, with another homozygous double recessive plant, small rr and small r 0, small r 0, with red flower and round pollen grains when they test cross heterozygous blue or purple long RRR RO plant cross ratio, they receive phenotypic ratio of 7:1:7 as the been illustrated here. Blue or purple long RRR RO all blue or purple long cross with red round, blue or purple long 192, red round 182, and blue or purple round 23, red long 30, means all progeny are 427. This is the linkage. Remaining part we will continue in the next period.